Hello and welcome to my channel. So in today's video, we are going to focus on biological membrane and its transport. So we will be focusing on biological membrane transport. We know that biological membrane is a semi-permeable membrane and it allows entry of only selective molecules. So today we are going to discuss three methods of transport of molecules across the biological membrane, out of which first we have diffusion, which is a passive process, does not require any kind of energy or transporter. Second, we have facilitated diffusion, and third, active transport. So the first process, that is diffusion, it is a passive process of transport in which the molecules move from higher concentration to lower concentration. They keep on doing so until the concentration is equal across a space. For example, if we open a bottle of ammonia in a room filled with people, the ammonia gas is at its highest concentration in the bottle. And it will start diffusing from the bottle so that it moves from higher concentration to the lower concentration. This type of process doesn't require any energy. And factors that affect diffusion are first, a substance will tend to move into any space available until it is evenly distributed throughout it. After a substance has diffused completely through a space, removing its concentration gradient, molecules will still move around in the space, but there will be no net movement of the number of molecules from one area to another. This lack of a concentration gradient, in which there is no net movement of the number of molecules from one area to another, is known as dynamic equilibrium. So the first factor which affects is steepness of the concentration gradient. How steep the concentration gradient is, that is, what is the difference between concentration across the membrane. The greater the difference in concentration, the more rapid the diffusion. So we can show it like this. If we have, this is outer membrane and this is inner membrane. So if the difference is too large, the molecules will tend to diffuse faster. And the closer the distribution of the material gets to equilibrium, the slower the rate of diffusion. So as it starts reaching near to equilibrium, the diffusion rate also decreases. Second step, we have mass of the molecules diffusing. If the molecules which are diffusing, they are heavier, so they'll move slowly. Therefore, they diffuse more slowly. Next, we have temperature. Higher temperatures increase the energy and therefore the movement of the molecules, increasing the rate of diffusion. Solvent density. As the density of a solvent increases, the rate of diffusion decreases and the molecules slow down because they have a more difficult time getting through the denser medium. If the medium is less dense, diffusion increases, and cells primarily use diffusion to move materials within the cytoplasm. Any increase in the cytoplasm's density will inhibit the movement of the material. Second, we have facilitated diffusion. Facilitated diffusion requires some transporter. So the materials diffuse across the plasma membrane with the help of membrane proteins, which are transporters. A concentration gradient exists that would allow these materials to diffuse into the cell without expending cellular energy. So this type of process doesn't require energy, but it just requires membrane transporters. However, these materials are ions or polar molecules that are repelled by the hydrophobic parts of the cell membrane. Since the outer membrane, we have phospholipid bilayer like this. So anything which is trying to enter inside the membrane, if the ions or polar molecules are trying to enter, they are repelled by the hydrophobic part. So more the hydrophobic a molecule is, more easy it is for the molecule to enter inside the membrane. Okay. Facilitated diffusion proteins shield their materials from the repulsive forces of the membrane and allow them to diffuse into the cell. So these proteins are called transport proteins, and they can be channels or carrier proteins. So now we have channel proteins. Channel proteins are transmembrane proteins that fold in such a way so as to form a channel or pore through the membrane, and each channel is specific for one particular substance. Passage through the channel allows polar compounds to avoid the nonpolar central layer of the plasma membrane that would otherwise slow or prevent their entry into the cell. So one example, we have aquaporins. These are channel proteins that allow water to pass through the membrane at a very high rate. This is protein channel, and these are molecules being transported. There are some channel proteins which are always open, but many are gated, and they can be opened and closed. So if a channel is ligand gated, the attachment of a particular molecule to the channel protein may cause it to open. Other channels are voltage gated, which require a change in voltage across the membrane to open them. Cells involved in the transmission of electrical impulses, such as nerve and muscle cells, they have voltage gated ion channels in their membranes. Next, we have osmosis. Osmosis is totally focused on water potential. So it is a process by which molecules of a solvent pass from a solution of low concentration to a solution of high concentration through a semi-permeable membrane. It is a passive process and happens without any energy usage. It involves the movement of water from high to low concentration. So if the concentration of water is high, high water, low solute. Low water, high solute. Which means if water is moving from higher concentration to lower concentration, solute will move in the opposite direction. Difference between osmosis and diffusion. Osmosis is a limited, which is only limited to the liquid medium. Diffusion occurs in liquid, gas, and even solids. Osmosis requires a semi-permeable membrane, whereas diffusion doesn't require. Depends on the number of solute particles dissolved in the solvent, and it depends on the presence of other particles. Only water or another solvent moves from a region of its high concentration to a region of its lower concentration, and any type of substance moves from area of highest energy or concentration to region of lowest concentration or energy in case of diffusion. There are three types of osmotic solutions: isotonic, hypertonic, and hypotonic. In the isotonic solution, it is one that has same concentration of solutes both inside and outside the cell. And in the hypertonic solution, it is one that has higher solute concentration outside the cell than inside. Because the concentration is higher outside than inside, that's why the cell shrinks. So see what happens now. The higher solute concentration. There is high salt outside the cell and less inside. So since this is osmosis, it has to move in the opposite direction. So water will be lost from inside to outside. So when water is lost, the cell shrinks. In the hypotonic solution, it is the one that has higher solute concentration inside the cell than outside. So since water is moving from outside to inside, that's why the hypotonic solution, the cells they're swollen. So in hypertonic solution, water moves out of the cell. In isotonic, no net movement. In hypotonic, water moves inside the cell. Next, we have primary active transport. In primary active transport, hydrolysis of energy-rich molecules such as ATP provide energy required for transport of molecules from lower concentration to higher concentration across the membrane. In primary active transport, we are just concerned of one molecule moving from lower concentration to higher concentration, that is against the concentration gradient. Since it is moving against the concentration gradient, it requires a push from ATP. On the other hand, we have secondary active transport. In secondary active transport, one type of molecule migrates from higher to lower concentration the release of the energy which is released during this process, it is utilized by another molecule. The released energy is used to transport other molecule from its lower to higher concentration across the membrane. This can be of two types, Symporter and Antiporter. So if two molecules are moving in same direction, it's called Symporter. If two molecules are moving in opposite direction, we call them Antiporter. Example for Antiporter, we have sodium potassium pump. In sodium potassium pump, there are three sodium molecules which are moving out and two potassium molecules which are moving in. So movement of three sodium molecules from higher concentration to lower concentration. So there is high concentration here for molecule A. The energy which will be released from this transport, that energy will be utilized by B. That's all about the video today. Thanks for watching.